Autocoids. Autocoids means local hormones, and the word autocoids is derived from the Greek words autos, which means self, and akos, which means drugs. These autocoids are called as local hormones because these act at the site or near the site where they are synthesized. The most commonly known autocoids are histamine, serotonin, and bradykinin. These substances do not have any therapeutic application, but their agonists and antagonists have some therapeutic applications. But before going to know about their agonists and antagonists, we should know about the synthesis, release, and secretions of these autocoids, and also their pathological and physiological roles, and also about their receptors. So let's now see about histamine. This histamine is a naturally occurring imidazole derivative. It is present in skin, mucosa that lines the gastrointestinal tract, lungs, brain, cerebrospinal fluid, and also bone marrow. This histamine is not only present in our body, but it is also present in some venoms and sting secretions of insects. Coming to the synthesis, this histamine is synthesized from an amino acid called as L-histidine by the action of the enzyme histidine decarboxylase, which causes decarboxylation, that is elimination of carbon dioxide from histidine, and then histamine is synthesized. This decarboxylase enzyme is present in tissues, mast cells, gastrointestinal tract mucosa, and certain actively growing tumors. The activators of this enzyme are gastrin, nicotine, and also stress and some kind of infections will trigger the histidine decarboxylase enzyme, which leads to histamine release. The inhibitors of histidine decarboxylase are also present. One of the mostly known inhibitor is alpha-fluoromethylhistidine, but this doesn't have any therapeutic application, but it is used as a research tool. Coming to the storage, the released histamine, the synthesized histamine is stored in mast cells in most of the tissues. In mast cells, it is stored as intracellular granules by forming complex with acidic protein and macroheparin. This macroheparin is a high molecular weight heparin. Coming to the blood, in blood it is stored in basophils. These basophils will morphologically resemble the mast cells, but these are not exactly the mast cells. Coming to the stomach, in the stomach these are stored in histaminocytes. In the brain, it is stored in histaminergic neurons. Release. The release generally happens with two mechanisms. The first one is immunologic release. In the immunologic release, whenever there is initial exposure to the allergen, let us consider this exposure as exposure 1. Here, IgE antibodies are released, and these released antibodies will form complex with IgE specific receptors which are present on mast cells or basophils. Let me repeat it. Whenever there is exposure 1, IgE antibodies are released, and these IgE antibodies will form complex with mast cells or basophils. In the exposure 1, there is only the release of IgE antibodies and antibodies complex formation with the receptors, but here there is no histamine release. Whenever the re-exposure to the allergen happens, then cross-linking of mast cell membrane bound IgE antibodies will take place. In the cross-linking, one antigen will bind with two IgE antibodies. These IgE antibodies are already bound to the surface of the mast cells during the first release. So to those antibodies, now the antigen will bind after the re-exposure. So after the re-exposure, the degranulation of cytoplasmic granules will take place and the histamine will be released. Not only histamine, here other allergic mediators are also released. Whereas this kind of release requires energy and calcium ions, and the histamine released in this type of release causes type 1 hypersensitivity reaction or immediate allergy. Coming to the second type of release, that is chemical or mechanical release, the chemicals that cause histamine release are morphine, succinylcholine, D-tubercularine, and also radiocontrast media, which is used for the diagnostic purposes. They will replace the histamine from heparin protein complex, which is formed in mast cells. Generally, the exocytosis will take place when these drugs are taken. During the exocytosis, this histamine heparin protein complex will exocytosize, and it is exposed to extracellular cations like sodium and calcium ions, and the histamine is released after cationic exchange with these sodium or calcium ions. Now let us see the list of the drugs or chemicals that liberate histamine. The drugs that liberate histamine are morphine, D-tuberculin, trimethophan, polymyxin B, succinylcholine, hydralazine, dextran, polyvinyl pyrrolidine, and some of the detergents, substance P, bradykinin, radio contrast media, and also compound 48 by 80. The mechanical phenomenon that will cause the release of histamine are the exposure to heat, cold, sunlight and X-rays, and also proteolytic enzymes like trypsin and chymotrypsin will also cause release of histamine, and some venoms of the insects or reptiles will also cause the release of histamine. After the histamine has performed its physiological and pathological functions, it must be metabolized. So when histamine undergoes metabolism, generally two enzymes will act on histamine. One is histaminase and one is imidazole N-methyl transferase. So when histamine is acted upon by histaminase, it will be converted to imidazole acetic acid. When imidazole N-methyl transferase act, it will be converted to N-methyl imidazole acetic acid. And these will be excreted in urine by forming complex with amino acid. Coming to the drugs that inhibit histamine release, the main drugs are beta-2 agonists. The main drugs are beta-2 agonists. They are adrenaline, ephedrine and isoprotenolol. And also mast cell stabilizers, which are disodium chromoglycate and ketotifen. This disodium chromoglycate is mainly used in asthma patients. And also negative feedback control will inhibit the histamine release. In this negative feedback control, no external drug will act as histamine release inhibitor, whereas the histamine that is released from the same mast cells and basophils, when it is exceeded its normal limit of release, it will act on the H2 receptors of both mast cells and basophils and cause negative feedback control, which will inhibit the histamine release.